Hello everyone, welcome in today's session, and today's session is about Chapter 13 Plant Growth and Development NCERT Exemplar. So let us start today's session. This is our disclaimer. Let us look about the learning objective of today's session. So we will solve the NCERT Exemplar question from the chapter Plant Growth and Development, and by solving these questions we can enhance our fundamental concept from this chapter, right? Let us have a look about the index part of today's session. So there will be some multiple choice question number 1 to question number 5. There will be some very short answer type of question from question 1 to question 6. There will be some short answer type of question 1 to question 9, and one long answer type of question. Let us start today's session with the first question. The first question is, ethylene is used for. This question is coming from the NCERT section 13.4.3, and the options are as follows. A. Retarding ripening of tomatoes. B. Hastening of ripening of fruits. C. Slowing down ripening of apples. And D option is says both B and C. Let us see the answer of this question. So the correct answer is option B. Ethylene is used for hastening of ripening of fruits. So ethylene is a crucial plant hormone, and we will explore some specific function of the ripening of fruits. So ethylene is used as a gaseous hormone, right? So this characteristic allows it to diffuse easily and affect the surrounding fruits, making it a powerful agent in agriculture. Ethylene ability to help ripen other unripe fruit, and it is a key feature of this particular hormone. Now, we can think of it as a catalyst that triggers a chain reaction, right? You can think like how the catalyst trigger the chain reaction. In the same way, this particular hormone comes from the ripened fruit and enters to the unripened fruit and make it ripe, that also. Some other functions. So other functions of ethylene, let us see that. Okay, next question is here. Coconut water contain. This question is coming from the NCERT section 13.4.3, and options for this questions are as follows. A. ABA, abscisic acid. B. Auxin. C. Cytokinin. D. Gibberellin. Let us see the answer. So the correct answer is option C, cytokinin, right? So coconut water contain which particular hormone? So it contains the cytokinin. Let us see the explanation part. Okay, so this coconut fruit is what type of fruit? It is a drupe fruit structure, and presence of the endospermic layer, that is the key to understand the natural composition of the coconut water, because it is having the endospermic layer which is having this coconut nutrient. It contains a lot of nutrient in that, in the form of coconut water. Now coconut milk as the liquid endosperm This coconut milk is actually the liquid endosperm that provides the necessary nutrients and hormones for the developing coconut embryo. And that is why it's rich in cytokinin. The presence of cytokinin in the coconut milk is very significant factor that promotes plant growth. It supports cell division, which is essential for the development of the plant. Now, the other hormones. The absence of other hormone in coconut milk emphasize the importance of cytokinin. Means this cytokinin is there and that enhances the division of the cell. So this unique composition makes the coconut water a valuable resource for studying the plant growth. Yes, if you want to study the plant growth, then you can take this coconut milk, and this is the very good resource that you can study about the how the plant is showing its growth, how the cells are dividing. So that features come from this coconut milk. Let us see our next question. The effect of apical dominance can be overcome by which of the following hormone? This question is coming from the NCERT section 13.4.3 and options are as follows. A. Indole acetic acid, abbreviated as IAA. B. Ethylene. C. Gibberellin. D. Cytokinin. Okay, so the correct answer that is the apical dominance and how it can be influenced by the plant hormone. Which plant hormone it influences? So that is the option D, cytokinin. So cytokinin is antagonistic to auxin, promoting lateral growth and overcomes the apical dominance, because the auxin is responsible for the apical dominance, but cytokinin, it promotes lateral growth, just antagonistic to the auxin. So what they do? They promote lateral bud growth, right? So cytokinin allows plant to develop the bushier appearance. The plant becomes bushy because of its lateral growth. Okay, so that can be beneficial in certain agricultural settings. So this ability to overcome the apical dominance is very crucial for the certain plant growth strategies, especially when a fuller plant structure is desired, right? Now, this indole acetic acid, or this is a type of auxin, right, that support apical dominance by encouraging the upward growth. Okay, the growth is upward and downward also, because the auxin is present at the tip of the shoot apex, whether it is a root apex or a shoot apex. So it is an important to understand how these hormones work together and against how they work against each other as well. And ethylene, while not directly related to the apical dominance, plays a significant role in fruit ripening, showcasing the diverse role of plant hormone. Now, gibberellin's role is to overcome the bud and seed dormancy. Highlights is important in the plant growth, right? And especially in breaking the dormancy for growth. So here you can see the lateral buds in the figure. Lateral buds are present, and when you cut the apex of the shoot, what happened? The lateral buds comes out from the axils of the leaves that are present at the nodal region. Okay. Next question, and this question is match the column type of question, coming from the NCERT section 13.4. Now, if you see the column 1, what are the items? Let us see that. A. IAA. B. IBA. C. Ethylene. D. GA means gibberellic acid, and E, cytokinins. If you see the column 2, then what are the items? First, herring sperm DNA. 2, bolting. 3, stomatal closure. 4th, weed-free lawns. And 5th one, ripening of fruits. So we are having the 4 different options, right? So you have to match the column 1 and column 2 correctly. The item should be matched correctly, and then you can see which of the options is correctly given in the 4 of the options. So you can opt that particular option. Let us see the answer of this question. So the correct answer is option A. So A, indole acetic acid, weed-free lawn. Indole acetic acid is used as a weed control. So this hormone is a key player in agricultural management. So 2,4-D, a synthetic indole acetic acid that kills the broadleaf weeds. Okay, broadleaf weeds, maintaining healthy lawns and fields. So this is a type of weedicide, right? Showcasing the practical application of the plant hormone knowledge. Now, next is the matching ABA, abscisic acid stomatal closure. Means this is a critical function for plant managing the water stress, closing of stomata. So, abscisic acid is a stress hormone. Means when the plant is in the stress, any type of stress condition, this particular hormone comes out. So, abscisic acid acts as a signal for the plants to conserve water by closing of stomata, which are the pores on the leaves responsible for gaseous exchange. Now, by shutting down stomata, abscisic acid reduces the rate of transpiration, helping plants to survive in a dry conditions. Next is ethylene. So, ethylene is a type of gaseous hormone. We all know that, right? And that is responsible for the ripening of fruit, leading to changes in the texture, color, and flavor as well. D is gibberellic acid, and gibberellic acid does the phenomena of bolting. So it is matched with second, column 2, second option. And gibberellic acid is responsible for the phenomena of bolting, which is when a plant rapidly grows taller, often before flowering. So what does it do? When gibberellic acid is spread on plants, it increases the internodal region of the stem and making it a plant appear much taller if you compare it with the normal plant. So, this rapid growth can make the plant extremely tall, which is why the gibberellic acid is often used in agriculture to manipulate plant height. Next, E, cytokinin, right? Isolated from the herring sperm DNA. So, cytokinin is a very crucial for promoting the cell division and growth, particularly from the plant roots and shoots. It is an important to note that the other options in the further question is incorrect, right? Let us see the next question. The term synergistic action of the hormone refers to. This question is coming from the NCERT section 13.4.3. Let us see the option for this question. A, when two hormones act together but bring about opposite effect. B, when two hormones act together and contribute to the same function. C, when one hormone affects more than one function. D, when many hormones bring about any one function. So what does it do? The correct answer is option B. When do the hormone act together and contribute to the same function? So this is the synergistic effect, right? Synergistic effect means the two hormones coming together and promoting one particular function. They are working together in that sense. So it increases the effect of functioning of each other, right? So it is when both the hormones coming together and performing one simple function. So it is promoting that particular function both the ways. And what is the result of this? There will be enhanced effect of both the hormones. You will see the effect. That effect is enhanced as both the hormone promoting for one particular function. Now, if you see the example, so cytokinin is antagonistic to the action of auxin. So there is a synergistic effect we see in the case of plant, okay, but there are some antagonistic effect as well. The cytokinin is showing the antagonistic effect of the auxin. Auxin is promoting the apical dominance, whereas the cytokinin lateral dominance. So other options like A, C, D are stating the incorrect statement. The correct one is given in the B statement, right? Okay, let us see the very short answer type of question. First question for this. Fill in the places with the appropriate word. This question is based on the NCERT section 13.1.4 and 13.4.3. So we are having the three statement and we have to fill the correct word in that fill-ups. A phase of growth which is maximum and fastest is dash. B. Apical dominance as expressed in the dicotyledonous plant is due to the presence of more dash in the apical bud than in the lateral ones. C. In addition to auxin, A dash must be supplied to culture medium to obtain a good callus in the plant tissue culture. Let us see the answer. So if you see the first statement, a phase of growth which is maximum and fastest is, what does this mean? It means exponential phase, right? When the growth is at a very fast rate, that means it is showing the exponential phase. Now in the next one, the apical dominance as expressed in the dicotyledonous plant due to the presence. So here both the progeny cells following the mitotic cell division retain the ability to divide and continue dividing. Next one, apical dominance has expressed in dicotyledonous plant is due to the presence of which hormone? Apical dominance is due to the auxin hormone, and they are present at the apex of the at the apex. Which apex? Shoot apex and root apex. Both the apex, this hormone is present, auxin, right? So auxin suppresses the growth of the lateral buds and promote the growth of the apex, shoot apex and root apex. So auxin always are produced in the apical bud, right? They are present at the apex. They are transported downward or the other parts of the plant, right? Including the lateral buds. So where the high concentration of auxin is present, it is present like it is present in the apical bud, but they are present in a very less amount in the lateral buds, and in fact due to the higher concentration of auxin in the apical bud, the lateral buds are showing their less growth or inhibits their growth. Now let us see the C1. In addition to auxin, cytokinin must be supplied to the culture medium to obtain a good callus in the plant tissue culture. So if you want to produce doing any tissue culture technique and you want to produce the callus, so you have to take the two combinations of the hormone. First is the auxin and second is the cytokinin. Now, what is the function of auxin? Auxin promotes cell elongation and root formation. And what is the function of cytokinin here? Cytokinin promote the cell division and shoot formation. So together they ensure the proper callus, uncontrolled mass of uncontrolled growth of mass of cells, that is called as callus, right? This callus is formed due to the presence of this both the hormone auxin and cytokinin. Let us move to the next question. Plant growth substances have innumerable practical applications. Name the plant growth substances you should use to. This question is based on the NCERT section 13.4.3. Now, let us see what plant growth substances we are talking about. A. Increase yield of sugarcane. For this, which hormone you will use? B. Promote lateral shoot growth. C. Cause sprouting of the potato tuber. And D. Inhibit the seed germination. Very important question. Let us see the answer of this question. So, gibberellin is used to increase the yield of sugarcane, right? So, it increases the length of the internode and the sugar is concentrated in these internodes, right? So result in increased yield and overall yield we get if the internodes of the sugarcane are increased. So it also enhances the sucrose accumulation in these stem part. Cytokinin promote the lateral shoot growth. So which plant growth substance they are talking? They are talking about the cytokinin, means the promote lateral shoot growth. For lateral shoot growth, there is a requirement of cytokinin. They break apical dominance. Yes, apical dominance is broken by this particular hormone and promote cell division and branching, right? This plant growth substances enhances the cell division and also the branching. Ethylene causes sprouting of potato tuber. This ethylene, this is a plant hormone that causes the sprouting of potato tuber. Breaks dormancy of potato tubers and also promotes their sprouting. Ethylene. Now, abscisic acid, very important plant growth regulator. This abscisic acid inhibit the seed germination, right? It acts as a growth inhibitor hormone. Okay, it do not allow the plants, or you can say the seeds, to germinate. It keep them in a dormant condition. So promote seed dormancy. Yes, it promote the seed dormancy. Do not allow the seeds to get germinated. So it helps seeds to survive for unfavorable condition and also provide protection. And it can be stayed for many years in the form of seeds because of this particular hormone. Next question. A primary root grows from 5 cm to 19 cm in a week. Calculate the growth rate and the relative growth rate over the period. This question is coming from the NCERT section 13.1.4. Let us see the answer. So, growth dependent on 3 factors. There are 3 different factors on which the growth is dependent. First thing is the initial size, represented as W0. Rate of growth, it is represented by small r, and the time interval, that is t. for which the rate of growth is retained. Growth rate is equal to the change in length upon time. And we can represent growth rate as W1, that is, after a time being, what will be the size of the plant, that is represented by W1. So W1 minus W0 divided by the time that is taken. So GR, a primary root grows 5 centimeter to 9 centimeter. So 19 centimeter is the W1 and W0 is 5. And what is the time period? 1 week. So 19 centimeter minus 5 centimeter divided by 1 week. So what you will get? 19 minus 5, that is the 14, right? 14 centimeter per week. This is the growth rate. So relative growth rate, let us talk about this phenomena, relative growth rate, because we have to find out this also. Now, for the relative growth rate, we have to use the formula W1 is equal to W0 e to the power RT, where W1 is the final size, yes, and W0 is the initial size of the plant. R is the growth rate, T is the time, and E is the base of the natural logarithm. So here, what we will take, R is equal to log W1 minus log W0 divided by T, because we have to find out the relative growth rate. So RGR, that is the log W1 minus log W0 divided by 1. 1 means 1 week. So it will be log 19 minus log 5 divided by 1. So it will be log 19 will, the value of log 19 is 2.944 minus 1.609. 1.609 is the value of log 5. Okay. So what we will get 1.335 per week. So growth rate, what is the growth rate? 14 centimeter per week, that we have already solved earlier. And relative growth rate will be 1.335 per week. Let us see the next question. Gibberellins were the first discovered in Japan, where the rice plants were suffering from the Bacne disease. This is a type of disease which is also called as foolish seedling disease, caused by the fungus Gibberella fusicori. This is the name of the fungus, Gibberella fusicori. Okay, this question is based on the NCERT section 13.4.3.2, right? So you have to give two answers for this. Give two function of this phytohormone, means gibberellin. And the second, which property of gibberellin cause foolish seedling disease in rice? So two functions of gibberellins are there. Let us see that. It produces the phenomena of bolting, very important. Growth of the internodal region just before flowering of the stem in the rosette plant. And second is the induce of seed germination, right? So here the breaks the bud and seed dormancy. The bud will break down and seed dormancy, induce seed germination. Rice seedling plants show excessive growth in their internodal region upon infection with the fungus. So the fungus, Gibberella fusiculae, it is having some phytohormone, and that phytohormone is gibberellin. Later it was discovered. So these hormone first discovered in Japan with the rice plant, which were affected by the disease known as Bacne or the foolish seedling disease, right? So this fungus produces excessive amount of growth because plant hormone gibberellic acid. It contain or produces the plant hormone gibberellic acid. So the plants became taller than the normal plant. Okay. So that is why it is called as foolish seedling diseases, right? So this hormone first discovered in Japan when the rice plants were affected by this particular disease. Okay. And the plants were taller than the normal plants. Many results into death of the plant. Okay. This hormone also causes the death of the plant. Next, gibberellins promote the formation of dash flowers on genetically dash plant in cannabis, where the ethylene promote the formation of dash flowers on genetically plants. This question is based on the NCERT section 13.4.3.2. Let us see the answer. What is the role of gibberellins? Let us see that. It promotes the male flower formation. Okay. So the first fill-up will be the male flowers and acts on genetically female plants. So genetically female plants. Second fill-ups will be the female plants. And that causes sex reversal from the female to male. Right? Formation of, it changes the sexes, that is, it can reverse the sexes, that is, from female to male. And what is the role of ethylene? So promote female flower formation, right? So ethylene promotes formation of female flower. The fill-ups will be the female flower on genetically plants. The third fill-up will be the female. So it acts on genetically male plant because it is also doing the reversal. It can cause sex reversal from male to female. So the gibberellin is causing reversal of male to female in cannabis, right? Whereas the ethylene causes reversal from male to female. Next question. A farmer grows cucumber plant in his field. He wants to increase the number of female flowers in them. Which plant growth regulator can be applied to achieve this? So they are talking about specifically for the cucumber plant, right? So this question is coming from the NCERT section 13.4.3.4. Let us see the answer of this question. Ethylene. What type of hormone it is? Gaseous hormone, right? It plays a major role in determining the sex of monoecious flower, right? So they are significantly impacting the yield by increasing the number of female flower, right? So it is associated with the promotions of femaleness in plant. If they are promoting the female flower to bloom more, then they are promoting the femaleness in plant, right? So what does the farmers do then? The farmers must spray ethylene hormone, right? So it will make more flower, more female flower, and from the female flower what develops? The fruit. In the case of cucumber, they will produce more female flowers, more cucumber. So cucumber crop to produce more female flowers. In that way, it increases the yield of the cucumber. So it will increase the yield. Next question. This question is a short answer type of question. Let us see the question first. What are the structural characteristics of? This question is coming from the NCERT section 13.1. A. Meristematic cells near the root tip. B. The cells in the elongation zone of the root. Let us see the answer of this question. A. Meristematic cell near the root tip. So, where these meristematic cells are present, they are present at the very near to the root tip. The cells of this regions are called root apical meristem, right? Root apical meristem. And they are in very active state of division. They are continuously in a division state. So, their number increases continuously. Okay, now the cells of this particular region, meristematic zone, what is the characteristic of this type of cells? They are very thin walled cells and they are having a lot of cytoplasm, dense cytoplasm, and nucleus is very prominent. It means large nucleus is there in the center of the cell. And what they do, they are having the ability to divide and redivide, right? So here you can see the zone of the meristematic activity. Continuously in a division phase increases the number of these cells. Cells in the elongation zone of the root. Now this region is situated where? Just above the meristematic zone. Okay, just above the meristematic zone is the region of the elongation. Now here what happens? The cells get elongated. The large number of vacuoles develop in this region, in the cells, and vacuole fuses, all the small vacuole fuses to become a one large central vacuole. So it is having a lot of cell sap in it. Whatever the excretory substance, whatever the extra water is there, that all get deposited in this center vacuole, and the nucleus which were earlier present in the center shifted towards the periphery. So the cells get elongated in this particular region. Next question. Does the growth pattern in plants differ from that in animals? And do all the parts of plant grow indefinitely? If not, name the region of a plant which can grow indefinitely. This question is based on the NCERT section 13.1. So let us see growth in plants. So we all know that growth in the plant is very different from that of the animals because they show indeterminate growth pattern, right? This is because they are having meristematic tissue which allow them to grow continuously, right? And this meristematic tissue are very crucial because they enable plants to grow throughout their life, and this is the different from animals. This feature is not present in animal where the growth is usually determinate type. Now these tissue are present in specific parts of the plant. Okay, they are not present everywhere. They are present at the specific parts of the plant. Where are they present? Apical. They are present at the apical, intercalary meristem, lateral meristem. 3 meristems are there. So these tissue has the ability to divide continuously. The apical meristem will divide continuously, intercalary will divide continuously, as well as the lateral, that also divide continuously. Okay, now they contribute localized plant growth. Okay, the plant growth is localized here. It's not like everywhere. Means the plant growth you can see at the apex of the plants, at the intercalary region and the lateral region. Now, all the parts of the plant do not grow indefinitely. Okay. The root apex and the shoot apex having the apical meristem, so they are keep on growing. Okay. And they contribute the height, axis of the plant. Now, let us see how the growth occurs in animals. They have limited growth period. Okay. After an age, their body do not show any growth sign. Next question. Explain in 2-3 lines each of the following terms with the help of examples taken from the different plants tissue. This question is based on NCERT section 13.2. A. Differentiation. B. De-differentiation. C. Redifferentiation. Let us see the answer. So first we will see the de-differentiation. It is a permanent in size. Means after the meristematic activity, the new cells are formed. They get differentiated to a particular type of cells. Means they are now in permanent size, structure, their composition and function. They perform one particular function. So they are present as a definite type of tissue. They are present in a definite type of organs, right? Now, if we talk about the example, the meristematic tissue in the plant gave rise to a new cells. And this cell get matured, it get differentiated and perform a particular function by forming a special type of tissue. So, cell distal to the root apical meristem forms the root cap. This is one of the example that showing the differentiation. And cells of the periphery means surrounding these meristematic cells, they are present at the periphery, forms the epiblema, cortex region, endodermis, pericycle and vascular. So here you can see the differentiation. Cell division forms the meristematic cell. Meristematic cell undergoes the elongation and maturation and then they forms the mature cell. So this is showing the differentiation. Now let us see the de-differentiation. So this is a despecialization of the differentiated cells. Means the cells once get despecialized can regain their ability to divide. Means the differentiated cells come back, they regains the activity of division. So this is a very crucial for the plant growth and regeneration, allowing plants to adapt and repair mechanism. So what happened? They can then become specialized again, ready to perform few particular functions. Okay. Now, if you see the examples, the stem of the dicot plant, like cortical cells, can undergo de-differentiation. And this is like a support staff member training become a manager, right? So, now the meristematic cell, if you think about the meristematic cell, they forming the cambium, which is essential for the plant growth. So it is like cambium is not present always in the between the two vascular bundle. There are the parenchymatous cells only, but this parenchymatous cell transform itself into, changes itself into, de-differentiated itself into the cambium cells, and it gets the activity of the meristematic cells. So here you can see the differentiation. Once the cell get differentiation, it comes back to differentiation and regains the previous cell characteristics. Next, redifferentiation. So the cambium cell formed from dedifferentiation again redifferentiate. So once they get differentiation, they do the activity of that differentiated cell, and again they come back to their original position. Means from secondary cortex cells, the secondary xylem and phloem elements formation. So phalloderm is again another example. Phalloderm is formed in the case of secondary growth of woody dicot plants, right? Next question. Auxins are growth hormones capable of performing cell elongation. They have been used in horticulture to promote growth, flowering and rooting. Write a line to explain the meaning of the following terms related to auxin. This question is based on the NCERT section 13.4.3.1. So let us see what we have to explain. A. Auxin precursors. B. Antioxidants. C. Synthetic auxin. So let us see. A. Auxin precursor. It is a raw material required for the formation of auxin hormone. Okay, the raw material that is required for making of the hormone auxin. Now, indole acetic acid, this is synthesized from which particular amino acid? Tryptophan. Okay, adenine compounds and the derivative of carotenoids. So these are the precursors of the auxin. Now let us see the antioxidants. Compound that inhibit the action of auxins are called as antioxidants, right? So if you see the examples, then p-chlorophenoxyisobutyric acid, that is PCIB. Okay, this is one of the example. Another is the TIBA, TIBA. These are the compounds that are working just stop the activity of auxin. So they are called as antioxidant. Synthetic auxin, some are the chemical based auxin. So they are synthesized not by the plants but artificially by human. So examples 2,4-D, this is used as a weedicide. Another examples, naphthalene acetic acid, dicamba, IBA, that is called as indole butyric acid. These are some of the examples of the synthetic auxin. Next question. The role of ethylene and abscisic acid is both positive and negative. Justify the statement. Let us see the answer of this question. So here we can see a tabular representation of the role of ethylene and abscisic acid. Okay, now let us see the ethylene activity first of all. Positive effect, it promotes the horizontal transverse growth. Okay, it is not showing the vertical growth, but it is showing the transverse or horizontal growth. Makes positive geotropic, means the roots goes downward. It is showing the positive geotropic phenomena. Promote apical dominance. Like we know that the apical dominance is given by the auxin hormone, but yes, ethylene is also promoting as a apical dominance. It breaks the seed and bud dormancy. Abscisic acid keeps the seed and bud as a dormant condition, but ethylene overcome this particular phenomena. Enhances fruit ripening and the root initiation. In some of the plants, it always promotes the root initiation, means formation of the roots, and also the fruit ripening. Okay. Now, these are the positive effects. Now, let us see the ethylene negative effects. Inhibit the longitudinal growth. It do not allow the growth in a height, whereas it allows growth horizontally. Decreases sensitivity to gravity. Root become apogeotropic, right? It enhances the senescence of the leaves, means aging. Aging phenomena occurs due to this particular ethylene hormone. Prolonged dormancy of the lateral buds. Lateral buds become dominant and it mediate formation of abscisic acid under the water stress condition. When the water is not available in a proper condition, then it mediate the formation of abscisic acid. Now, let us see the abscisic acid positive and negative effects. Positive effect, first of all, abscisic acid, it is used as anti-transparent. Anti-transparent means it allows the stomata to get close and reduce the rate of transpiration, right? So when there is a less water is available in the field, this abscisic acid works there and it is working as an anti-transparent. It induces the flowering and root initiation in some plants. Okay. And prolonged dormancy, used for the prolonged dormancy for the seeds, for the buds, right? Increases resistance of plants to cold and other types of stresses when there is a stress condition. Okay. So this particular hormone is secreted at the time of cold, at the time of like water stress condition, and that is why it is known as stress hormone. Negative effect, promote abscission of flower and fruits. Okay. It promotes the abscission layer formation in the case of flowers and fruits. So they get detached from the plant. Stimulate the senescence of the leaves, means it also causes aging, stopping the protein formation and RNA synthesis. It promotes the bud and seed dormancy, right? And act as an antagonist to the gibberellin, just opposite. It function as gibberellin. So it counteracts the effect of other growth hormone like indole acetic acid and cytokinin. It also inhibits the seed germination and growth of embryos in the seeds. So this is all about today's session. Thank you very much for attending today's session.